So in this study, we wanted to study Machado-Joseph disease or spinocerebellar ataxia type 3, a disorder that we have been studying for many years. And we know that this disorder is caused by a mutation that involves the overrepetition of the trinucleotide repeat CAG. And this translates into a polyglutamine tract within a protein designated as ataxin 3. So this mutant ataxin 3 with an overlong polyglutamine tract will cause neurodegeneration in different brain regions. Now, patients will experience trouble to walk, to articulate the speech, and many other symptoms, particularly motor symptoms. But what was less explored and that we wanted to investigate in this study was the non-motor symptoms, and particularly the circadian rhythm disruption. These circadian rhythms are the internal 24-hour clocks that govern both the temperature, the sleep, and the overall physiology of each one of us. Now, the different neuropathological events of Machado-Joseph disease have been comprehensively explored over the years, but not the circadian rhythm disruption. And so we wanted to study this here, and particularly we were interested in using a translational approach where we investigate this both in patients, in animal models, and in vitro cell models. And by doing this, we expect, and you see in the paper, that we unraveled this circadian rhythm disruption in Machado-Joseph disease, potentially in other spinocerebellar ataxias, and also a little bit of the mechanism that is involved in this disruption of the circadian rhythm that can also be very important in terms of therapeutic approaches in the future.
We conducted a clinical assessment of the rest activity rhythms in MCD patients using actigraphy and identified several marked circadian alterations. With the clinical progression of the disease, MGD patients show a decrease in the robustness and stability of their rhythms, along with a higher fragmentation of their activity profile. They also displayed increased nocturnal activity, reduced sleep efficiency, and shorter total sleep time. To understand the neurobiological mechanisms behind these dysfunctions, we studied a YAC-MGD transgenic mouse model encoding the full mutant ataxin 3 gene. Our initial question was whether circadian activity will also be affected in this model, and so we performed wheel-running experiments to continuously record their activity. MGD mice exhibited reduced activity levels in a specific part of the day, increased behavioral fragmentation, and required 3 additional days to re-entrain after a jet lag protocol compared to controls. To understand if other biological outputs of circadian rhythms were affected, we implanted telemetric data loggers in mice to monitor their core body temperature. MGD mice showed disrupted temperature rhythms, including a phase advance and an abnormal increase in temperature at the onset of the active period. These results indicated a profound dysfunction in the central master clock, located in the suprachiasmatic nucleus of the hypothalamus, responsible for synchronizing time across the body. Immunohistochemistry analysis revealed decreased levels of 2 neuropeptides crucial for the robustness of this system.
Given the importance of central clock synchronization for the circadian rhythm system, we assessed the mRNA levels of 4 clock genes across the day in different brain regions. And we found, so the results showed, that a significant reduction in the expression of levels of BMAL1, clock, PER2 and CRY1 at a specific time point of the day in the cerebellum of the MGD mice. And the cerebellum is relevant because it's the region heavily affected in the disease. The dysfunction that we observe in MGD mice suggests that mutant ataxin 3 appears to interfere with normal clock regulation. In fact, we discovered a new function of wild-type ataxin 3 in activating the promoters of 2 clock genes, the BMAL1 and PER2, whereas mutant ataxin 3 loses the capacity to drive PER2 upon polyglutamine expansions. This study provides the first evidence that clock regulation is markedly affected in MGD patients and also in experimental models of the disease, in vitro and in vivo models. Understanding clock dysfunction in MGD is crucial not only to better understand MGD pathophysiology, but is also relevant for the identification of biomarkers to monitor disease progression and also to test circadian-based intervention to tackle this devastating disease. Lastly, our study reinforced that circadian rhythms must be taken into account when we do research in neurodegenerative diseases, and also for the clinical studies, including for MGD and other ataxias.